Okay, well, good afternoon. So I will just go ahead and take over, and we will talk again about statistical issues, and primarily statistical issues on the quantitative side, right, as opposed to identifications. But the identification issues will come back to us in that as well. And so just to remind you where we started on Monday when I was talking about experimental design, is the motivation for the whole conversation is that we have a lot of variation and uncertainty which comes from various sources, and the main source of variation and uncertainty is the biological person-to-person, sample-to-sample, or within the same person, time point to time point variation. And so statisticians contribute to conduct effective and reproducible research. So what I also like to do now is to kind of emphasize that when we talk about reproducible research, it's actually not a thing, it's a spectrum, that there is a whole continuum of reproducibility, and different tasks may be held to different reproducibility standards. And so on one side of the spectrum, we have research which is publication only, which is not good, and unfortunately it happens enough, much more than we would like, right? So our goal is to move away from that. And then if we think specifically about data analysis, so there is repeatable data analysis, where if we have an analysis published, we want to be able to go back to the same data, repeat the same analysis steps, and get the same numbers back. And it is actually quite challenging by itself already. But it's not the only thing, because what we also want to do is to repeat the analysis, but maybe using methods which are the same in spirit, but maybe not exactly identical.
It's 9 o'clock.
Sorry. But using the methods which are the same in spirit, but not identical numerically, right? So many methods, they rely on tuning parameters or some secondary decisions which are required, but they're not necessarily the core of the method. And so we don't want our biological conclusions depend on those tuning parameters too much. And so ideally we will have a reproducible data analysis where we will use a slightly different variation of the tool or a different tool, and we would get qualitatively the same conclusion, so that our conclusions do not depend as much on the tools as they depend on the actual biology, right? That's really the goal. And this is on the computational side. And then also on the experiment side, what we also want to do, we want to reproduce the same experiment. If you take the same biological material, we should be able to redo all the data acquisition steps and data analysis steps. We will not get the same numbers back, but we would want to have qualitatively the same conclusions. And of course, the gold standard for reproducibility is to take new biological material, new patients, right, or new samples, repeat conceptually the same type of data analysis, and get qualitatively the same conclusions. And so what we try to do in my lab, so we are statisticians, we develop methods for experimental design, tools for data analysis. We try to contribute throughout the scope of this spectrum, and what we talked about on Monday contributes a lot to this. So how do we design experiments where we can avoid bias, minimize confounding, right, minimize variation. What I would like to talk about today is more on this side. So once we have an experiment, how can we make sure that we can first repeat the data analysis again and get the same numbers back, or how we can analyze the data in different ways and maximize the reproducibility of the conclusions. So that's really my goal. And what I would like to tell you first is to talk about MSSTATS and then different evaluations, and then also some of the extensions for that. So this was the motivating example long time ago, in fact, a collaboration with the Abersold lab at ETH way back, where one of the Abersold lab members was interested in comparing two breast cancer cell lines, low and high invasive, and the cell lines were exposed to different amount of oxygen, so there's normal oxygen and low oxygen, and the exposure was done for different time periods. And then within each of those combinations, we had multiple cell cultures, and for each cell culture, we had multiple technical mass spectrometry runs. So it was a pretty complicated experiment, and the collaborator was interested not only in comparing low versus high invasive cell line, but there were questions such as, is the change in protein abundance that follows change in oxygen exposure the same in both cell lines? or does oxygen exposure affect different cell lines differently? So these are kind of more complicated questions, and at that time there was really not much infrastructure to deal with this type of complex designs. And so what the colleague was doing, she was literally extracting some runs which kind of corresponding to some of the questions and doing some by hand Excel style manipulation with them. So it was very complicated, and we felt that we could do better than that. And so this is one summary of the full experiment, of which we look, where we look at all sample preps, all cell cultures, all replicates. And this is just one protein from this experiment. Each line here links the mean log intensity of one peptide. So this protein has 3 peptides. Each point is the replicate across all the conditions here. And so you can see that some things are quite easy. So clearly here, the low invasive cell line has the protein in higher abundance than high invasive cell line. So there's a big change. However, if you start looking at the difference or the effect of oxygen exposure, things become much less clear, right? And there's some missing values here and some outliers and so on. And so the motivation, the original motivation of our work was to come up with statistical methods which will take as input the output of data processing tools such as Skyline or DIA Empire or anything else, and develop statistical frameworks which can work with complex designs such as this one, handle outliers, handle missing values, handle complex comparisons, and provide approaches which are repeatable in the sense that there are scripts which you can repeat and you will get the same numbers, but hopefully also reproducible, meaning that they don't depend on the choice of the data processing tools too much. And that was the original motivation. And since then it grew to a lot of things. So now we include many different workflows. So in particular, we can work with data-dependent acquisition, targeted SRM with or without labeled references, and data-independent acquisition. In many ways, these experiments have the same type of data structures, but also in many ways, the data structures are quite different. For example, SRM may have labeled references, DIA will have a lot more features, and many of them can be noisy. DDA may have more missing values, and so we had to make sure that these methods work across the board, across different types of acquisitions. And so now the status is that we work with kind of the main objective, not the only, but the main objective is essentially find proteins that change between conditions, right? So what is changing? We really work very hard to accommodate arbitrary complex experimental design. So any combination of conditions, time points, and so on. Originally, it was designed to work with what Brendan would call chromatography-based quantification, so where we would integrate the chromatographic peaks, so DDA, SRM, and DIA, and PRM. However, in the last 6 months, we also released an extension to MSSTATS, which works with TMT labeling. So now we also work with chemical labeling. And so label-free or label-waste. And the functionalities that we would provide is data visualization. We think it's very important to look at the data. We hope it will not be the first time when we look at the data. You would look at the data already with Skyline or with other tools, but what we provide is somewhat complementary, as you will see in a minute, and statistical modeling and model-based conclusions. As I mentioned on Monday, sample size calculation, where we treat each data set immediately as a pilot study for something else and project the sample size for future studies. And also more recently, we developed tools for kind of making sure that our experiments are fit for purpose. So we have tools for assay characterization, and we have tools for system suitability and quality control. And I will mention this briefly as well. And let me tell you upfront that everything we do is free and open source, and we develop things in R. And you know that R is very popular in the statistics community. That was one of the original reasons, but I think there are more reasons than that. So from the user perspective, for the scientist or researcher perspective, we find that it is really easy, right? So it's lightweight, minimal software or hardware requirements. You can do data exploration on a laptop, and since we take as input process data, these are relatively small files in many cases, so it's actually quite easy to deal with. For us as developers, it is really nice because we rely on a very broad community of statisticians who can contribute technical libraries, you know, for different aspects of things that we need to rely on, which means that we're not a very large group, we're a fairly small group, but we can leverage the developments in the statistical community and bring them quickly to the community of mass spectrometry users. So to us, it's quite valuable, but also because it's a scripting language, it is also can be customized, can be extended to other needs. And also if you, for example, operate a core facility and you have certain pipelines, this type of software can be very easily integrated in the existing software pipelines. So that's another use of this. And I think most importantly, we like to work with scripting tools and with R, because we think that it contributes to good science. As I was mentioning earlier, so your script is your best documentation, because it requires you to say exactly what you did everywhere, and we find that this is the best way to document the workflow. And of course, things being open source, it means that you can open them up, you can look at what we do, you can modify them, you can criticize them, you can say you don't agree with us, and that's how things are, right? So we think this is a good way to go. So that's really the introduction to what we do with R. And so now let me just show you the outline of the data analysis workflow and how statisticians would see that. You probably have seen presentations by experimental scientists where they will talk in great detail about data acquisition and instruments and sample prep, and then they will have a little box bioinformatics, right? So I'm kind of doing this the opposite, right? I have this little cloud saying wet lab, right? The point is that there's quite a lot of statistics that has to happen before we get to the point where we collect the data, and also after when we collect the data, right? And so let me first kind of focus on this part, and then we will come back to the first parts. So the first thing is the experimental design, and we talked about this already quite a bit. So we have to understand what the goals are. In particular, MSSTATS is designed primarily for detecting changes in abundance, right? So does our experiment, biological question, does it call for changes, for comparisons of averages, or maybe it calls for something else? So MSSTATS can also be used for other goals. I will mention this briefly later. And of course, selecting conditions and replicates and technological aspects and so on. So we talked about this quite a bit. And so then, the actual experimental design, for example, this one, as I mentioned, it is an example of a factorial experiment. Factorial means that we have different factors, cell lines, oxygen exposure, and time. So there's 3 factors, and we have all combinations of these factors present. This would require certain type of statistical analysis. So this is another example also in kind of older collaboration. This was a study of osteosarcoma, so this is a childhood bone cancer, and our collaborators had 29 control subjects at a single time point, and then they had 14 subjects with the disease, and they actually had a temporal measurements for the subjects. They had before treatment at different time points at chemotherapy, before surgery, chemotherapy after surgery, and then they also had some time points much later as a follow-up. And so their questions were also fairly complicated. For example, which proteins are differentially regulated between controls and disease at diagnosis, but they also wanted to know which proteins in time came back to the levels comparable to the controls and which proteins never came back. So again, we tried, and this would require different type of statistical analysis because there is a time course aspect of this, meaning we have on the same person, we have multiple measurements, and it has to be analyzed differently. And so MSSTATS will work with all of that. And what we actually do, we will figure out behind the scenes what type of experimental design this is, and we will find the right model for this experimental design. So from your perspective, you don't need to worry about that. The only really important constraint that we have, or convention, is that in MSSTATS, each biological replicate has to have a unique identifier. So if you have person 1 in condition 1, and then you have person 1 again in condition 2, then we know that this is a paired design or a time course design. Otherwise, different groups will have to have different person identifiers. And so this is where we will know, we will essentially infer what type of design you have and how to deal with that. Okay, so that, and then the data processing. And this is where we don't do much. We truly rely on collaborations with many wonderful labs, such as the Skyline team and Alexei's lab, the Empire, Biognosis, OpenMS, max quant, progenesis. So we try to be essentially as inclusive as possible and not restrict your choices of data processing tools, because you may have good reasons of why these tools are used. And so the steps that we would do first is, well, loading the data and then visualizing the data in quality control, normalization, and then we will have to work extensively on missing and outlying peaks and quantifying protein in the run and looking for differentially abundant proteins. So this would be the next step. So this is an example of an input, and tomorrow you will actually have a hands-on session with MSSTATS. So I hope that you will be able to try that. But just to kind of clarify that our input is essentially an Excel spreadsheet, right? And the convention is that one row in the input is one LCMS peak, so one precursor, and or actually either precursor if it's DDA, or one fragment if it's DIA. So essentially one row corresponds to one peak intensity. That's what I'm saying, right? So here, for example, we have a protein. It has multiple peptides. We may have precursor charge, fragment charge, labeling if it's heavy or light if it's labeled, or just light everywhere if it's label free. And then we have conditions, biological replicates, runs, and then intensity. So essentially this is what statisticians call a log format, where you have one row per intensity of the peak, and then we will work with that. How do we get this Excel spreadsheet? Well, it depends. For example, Skyline exports a report in the MSSTATS format, so it will actually take the Skyline document and create this directly. Other tools don't necessarily do that. So for example, with MaxQuant, we have a converter. From many other tools, we have converters, which are essentially R scripts, right? So that you just take the files and we just run some R scripts on them to create the files in this format. Okay. And then after that, we would take a look at the data, do data visualization and data processing. So a very important aspect of data processing is normalization. And I would argue that probably the majority of, most of the time that we spend when working with new data sets is figuring out the normalization, because it is not obvious at all. And so let me just give you some examples. So here's the experiment, which is a series of yeast samples, and they were subjected to a stress. And so here the y-axis is the log intensity of all the features across all the proteins which were quantified for these samples in the run. So we have vertical lines separate time points, and each box summarizes log intensity of everything in this run. So we have 3 replicates per run. And so now if we look at the log intensities of every feature, we see some kind of trend upward. And the question is this the biological trend or is it a technological trend? And the answer is that it can be both, right? And it depends on many things, right? So for example, if it's a targeted experiment where we target a small number of proteins which are likely to be involved in response to the stress, it may very well be biology, right? If it's an experiment which is global discovery and the majority of things are not going to change, it may be some kind of experimental artifact. And so in the first case, we don't want to normalize this away. In the second case, we actually want to make sure that we eliminate that. But this is the type of plot that MSSTATS would actually make. Now, in this particular experiment, turns out it is a targeted experiment. So we have labeled reference peptides, which were spiked for each endogenous targeted peptide of interest. And so now MSSTATS will make a plot like that, where you have the labeled references separately and the endogenous peaks separately. And so now it is very clear that those references, they were spiked in as constant concentration as possible, so they don't have any biology. They're supposed to be constant. So there's some variability here, but nearly not as much as here. So clearly there's many of these changes are biological changes and not technological changes. And so in this case, what MSSTATS will do is to apply some type of normalization. So in this example, it's a quantile normalization, where we literally equalize completely the boxes across all the reference runs. And this is appropriate because these are reference labels, right? Labeled references, so they don't have any biology. We learn these shifts for each run. We apply these shifts to every endogenous feature. Now we see that clearly these proteins were upregulated in later time points. So quantile normalization is often used, but we really don't advocate to use it for anything beyond this type of situations when we have many labeled reference standards. So just in terms of the algorithm, so we order log intensity in each run, we calculate the average of each quantile across the run, substitute the log of intensity in each run with the average, and rearrange log intensity in the original order. So this algorithm has been developed originally for gene expression microarrays. And it is good because our normalization is very stable because it relies on many data points. So those averages of quantiles in each run, they're very stable. They account for many things potentially, right? So this is good. However, if we apply them to endogenous features and not to reference standards, then we run into the problem. So this type of normalization will assume that the majority of the proteins are not affected, and this can be way too aggressive. And so usually we use it only to normalize multiple standards. Say this again, what if what?
What happens if the effects that you're trying to correct for are not uniform? Maybe some peptides are affected or some individual.
Well, so it's okay. So it's a complicated thing, right? So let's say you have a label-free experiment, right? And you just have some number of standards that you try to use to normalize. You have no choice but to assume that whatever happened to the standards also happened to the endogenous features of interest, and you use exactly the same shifts that you apply to equalize the standards. You use the same shifts and you apply them to the endogenous features. Now here, in this particular example, we can do better than that because we have a standard spiked, literally, there's a reference counterpart for every endogenous peptide of interest. So then what you can do is to just do this normalization essentially for purpose of visualization or quality control. But you can do better than that because at that stage of statistical analysis, you will account. I will not talk much about that because I'm kind of focusing on DIA, but I can tell you maybe offline later that MSSTATS extends statistical analysis to account for those peptide-specific interferences if you have a labeled reference matching every endogenous feature as well. But in experiments such as DIA experiments, it may be tempting to use quantile normalization on the endogenous features. And it makes a really big assumption that the majority of the features are not changing between conditions. Otherwise, you will essentially normalize away some of the true biology with that. Okay. And of course, if you have a targeted experiment with only few features, then it's really not a good idea, right? Because then assuming that the majority are not changing is not a good assumption. And so here's the standard-based normalization. This is another normalization implemented in MSSTATS, where essentially what we're trying to do is to equalize the medians of the standards and then apply this to these boxes. And so you see here that after the normalization, the boxes became a little bit more constant, but they're not exactly constant because what happened to endogenous features is not exactly identical to what happens to the standards, right? And so the algorithm is that we subtract the median of log feature intensity of the standard and back the median of the medians, and we apply the same shifts to all the endogenous features. So the good point is that standards do not have biological variation. They are not dependent on the number of features. The bad thing is that they only count for deviations which occurred after we spike the standard. And the standards can be noisy themselves, right? So if we have only one standard peptide, it can itself be subject to interferences, and then we introduce more noise, then we actually normalize away. So usually, if we cannot have labeled or standards for every endogenous feature, which is usually the case in DIA, what we advocate for is to use at least more than one standard for normalization. And so what we would do then is to take maybe some standards for normalization, to learn the shifts, right, and apply these shifts to all the endogenous features, and also apply these shifts to the second set of standards, and then see how this normalized second set of standards behaves, right? So if the second set of standards became more stable, it means that normalization worked as expected. If your second set of standards became more noisy as the result of normalization, then it's an indication that the normalization essentially failed and introduced more noise than that cleaned the data. So that's how we like to use that. Okay. And also a very popular normalization, this is what we end up doing, for example, in DIA, is to use median normalization, where we only equalize the medians of the endogenous features across runs. But as I said, that it's similar to quantile normalization. It assumes that the majority of things do not change. So here's an example of a dilution series without labeled references. So clearly normalizing the medians removes away the dilution signal, right? So this is a really bad idea here. And so here's the algorithm where we subtract the median of all endogenous features and back median of the medians. So that's more stable than a single standard, assumes that most things do not change. And this, in our view, in absence of standards, in large-scale experiments such as DIA, this is our go-to normalization. Just so not equalizing the entire boxes, only equalize the medians because it's less aggressive. And in large-scale DIA, it's probably more likely that only a small percentage of features changes. So essentially, to summarize, in DIA, we would use, in absence of standards, we would use median normalization. If we have standards, we would use those as well. In SRM, if we have more labeled references, then we will work with labeled references and include this as part of statistical analysis as well. And just to repeat, a lot of things downstream can be automated. This is the part where we really need to think about how the experiment was done and what were the batch effects and potential artifacts which we may attempt to normalize. So this is a really important thing. The good news is that MSSTATS offers multiple options for normalization. You can look at these box plots before and after and have a sense of how things worked. So, and then this is really where the core kind of statistical contribution starts in terms of statistical analysis. Let me just give you the upshot. At the end, if we have two conditions of interest, I don't know, healthy disease or something like that, we would summarize our results in this form. So this is a volcano plot. The x-axis is log of the fold change. The y-axis is the p-value adjusted for false discovery rate, adjusted for multiple testing. And then we take negative log just so that significant values are large numbers. And we would use the horizontal cutoff. This is statistical significance, meaning that everything above this cutoff has the false discovery rate controlled at that level. And we can also add, after that, vertical cutoffs, which we call practical significance, where we say, you know, for whatever we do after with this data, below fold change of 2, we're not interested, right? So then we just, and this is totally fine as long as it's in addition to statistical significance and not instead of statistical significance. So this is if we have two comparisons, but oftentimes, for example, this yeast experiment I just mentioned with 10 time points, we have more than two comparisons. Like in a time course, maybe we want to compare time 1 to time 0, time 2 to 0, 3 to 0, and so on. So then we also make this type of heat maps. Here, blue means significantly downregulated, red means significantly upregulated, black means no change. And so the y-axis are all the proteins, and this is all the comparisons. And you can kind of start seeing the patterns, what goes up, what goes down through this. Okay, this is the conclusion. But now let me tell you what happens between data processing and this, right? So how we actually get to this point. And as I mentioned, we really try to be inclusive and make sure that we work with different data processing tools as much as possible. And in the beginning, we were worried a lot about formats and converters and just making it happen, you know, taking files in one format and actually read them into MSSTATS and make sure that things work. But very quickly, we realized that there is more to it than the formats. Because what turns out is the different tools, and you have seen already between Skyline and DIU Empire, and you will, Spectronaut as well, right? So, and many others, different tools, well, they're developed by different labs and they make different decisions for good reasons, right? Because they have sometimes different purpose, sometimes developed on different data sets, sometimes the developers have certain vision and certain problems are most important to them, right? And but all of that should not really affect our biology too much, right? We should be able to use different tools, which are all reasonable and, you know, and validated, and get the same type of biological conclusion in the end. And didn't quite happen that way. So we saw that our conclusions depended quite a bit on the input that we take, so on the data processing format. And we try to look into that more and see what exactly is happening. And so this is just some examples of that. So this is one data-dependent acquisition data set. So this was from Jürgen Koch's LFQ paper, 2014. And this is the DI data set from the Bruder paper that Alexei mentioned this morning. So these are both controlled mixtures. So what we did for them, we processed them with different data sets. And by we actually mean we went out to the developers of the tools and we asked them to process them to make sure that we did not misuse the tools somehow or didn't have wrong parameters and so on. So this is the processing by Skyline, MaxQuant, and Progenesis for DDA, and this is two versions of DIA. So let's just take a look at the DDA first, because we have three tools. So here the x-axis is the log intensity of everything that the tool reported in this experiment. So across all features, all peptides, all proteins, all runs, right? So just anything we have in this whole input file, we make a histogram of. So these are the histograms from the three tools. And so the first thing you see that these histograms do not look the same, right? So here, for example, Progenesis has range of values 10 to 30, MaxQuant 20 to 34, right? And Skyline has kind of broader range a little bit. So already numeric values are different, which by itself is not a problem, right? Because we're not interested in numeric values of the intensities, we're interested in their relative comparisons, right? And so if, for example, you integrate the peak entirely versus you truncate the tails and integrate the center versus you take the apex of the peak, you will have different numeric values for that peak, right? So it's not a problem as long as relative quantification is preserved, right, between the runs. So by itself, it's not a problem. Now, let's look more. I hope you can see from here. So for Skyline reports 203,000 peaks. MaxQuant reports 224,000 peaks. Progenesis reports 56,000 peaks. So order of magnitude less, right? At the same time, if you look at zeros, MaxQuant has no zeros. Progenesis has 600 zeros. Skyline has 1,300 zeros. So again, different decision in terms of what is zero or very close to zero values. On the other hand, let's look at the missing values. Skyline has 33 missing values. MaxQuant has 38,000 missing values. So so a lot of variation. And generally speaking, what happens is when you have very tall, well-resolved peaks, you will have difference in numeric values of peak intensity, but it will be more or less okay, right? The real difference between the tools happens in the noise level, right? When the peaks are very small or undistinguishable from the noise or absent completely, right? And also sometimes when we have interferences. And also, yeah, so these vertical lines, by the way, these are the counts of values which are either exactly numerically zero or very close to zero. So you see that MaxQuant clearly applies some internal thresholding here, right? So below 20, there is nothing. And then Skyline and Progenesis do other things. And same thing for DIA, right? So if we now look at Skyline versus Spectronaut, very different range of values, right? Number of peaks, 3 million here, 2 million here, 2,000 NAs in Skyline, 13 NAs in Spectronaut, right? But then different numbers of zeros as well. So clearly these decisions are apparent, right? If we just look at the summary of the data. And so by working with that, and it really took our group a very long time, and every time there's a new tool, there's a new kind of patterns, right, and new types of decisions. How can we handle all of that? And so the decision that we made fairly early on is to essentially not take the outputs of these tools literally, but try to read between the line and understand what was the intention of these tools. And again, when peaks are high and well resolved, it's okay. So especially in the low noise level, so what happens then? And essentially what this means is that different tools use different devices to express the fact that the peaks are low or low confidence or hard to quantify, right? And sometimes zeros, sometimes NAs. Sorry? Not yet, I will get to that, yeah. So, so we try to essentially not interpret this literally, but try to understand what the intention behind these values is. And so what we decided is to say, okay, when peaks have low values, we essentially don't trust them. Call them NA, 0, small values, no matter what, we will not trust these values. And by low values, essentially, we came up with this vertical threshold, and it's completely empirical. So the way we decided, so those histograms in many data sets we looked at, they're roughly symmetrical, more or less. So what we did was we took the median, we look at the 97th quantile to the right, calculate the distance from the median, got the same distance to the left of the median, and this is our threshold. So just looking at the symmetry, because the right tail is much better defined, and we said that below this vertical line, we will not trust any value from any tool. And sometimes it's 0, sometimes it's small value. MaxQuant had nothing, but it had missing values, right? So we have to deal with that. And so now the next thing we will assume that if things are in this region or missing, it's primarily because the peak, essentially the analyte was below the limit of detection or below the limit of quantification, and we could not quantify it reliably. The only exception is Skyline, because Skyline actually makes a distinction between things missing for reasons of low abundance versus missing for other reasons. So for example, if you have peaks which are highly overlapped, or in SRM you have a scheduled transition, but it's kind of got missed and you only see a portion of the peak, but not the entire peak, you cannot quantify this. Skyline reports NA to say, I cannot reliably quantify the peak, but the peak is there. For everything else, it will be value 0 or very close to 0 value. And so in the special case of Skyline, we leave NA untouched. In every other tool, we did not see these distinctions, and therefore we say if we have 0, NA, small value, all of this, we assume that the analyte is below the limit of quantification, and this is the definition in statistics of censored values. So censored means that it's missing, but it's informatively missing, meaning that we know that it's not arbitrary value, but it's supposed to be a small value. We just don't know what this value is. And turns out that, and so now the censored values will be the values which we count below this vertical line, which is defined by ourselves. And so the good news is that statistical literature has a lot of ways to deal with censored data. And we are relying on that to essentially accommodate missing values which occur in that way. So this is the representation of the data for one protein, and this is the abstraction that we use inside MSSTATS. So for one protein, well, first of all, the columns are the mass spectrometry runs. And the runs may be technical replicates of a biological replicate, right? So multiple runs are grouped by a subject. Some other runs are grouped by another subject, and so on. And the subjects are grouped by condition. And if we have the same subject and multiple conditions, we know it's a time course. If we have different subjects and different conditions, we know it's a group comparison design. And essentially, and this is the pink part, expresses the biological aspect of the experimental design and sources of variation and so on. And this is what gives us the answer ultimately to the biological question. But the rows are the technology, because we do not quantify the protein, we quantify in DDA peptide ions, in DIA we quantify fragments of the precursors, right? And so for us, feature is essentially a peak. So for example, if you have a protein with 2 peptides and 3 fragments per peptide, we have 6 features. So we just have 6 features listed in this table. And so the yellow aspect of this table reflects the technological aspect of the experiment. And Y's here, these are log intensities of peaks, and NAs may be occasional missing values which are reported by the tools. And so now we'll try to integrate this type of view, that ignoring low intensity values, with this type of representation of biology and technology. Well, so the first thing is, and again, we're relying on very kind of large statistical literature in this field. So this type of two-way tables, they can be represented with linear mixed effects models, which are very flexible in nature. They can express these different complex designs I was talking about, different types of features and so on. So this is one fairly traditional way of representing this. I don't want to kind of give you too many technical, it's getting pretty technical here. What I will tell you, and this is quite important, that this is type of models which are standard for this type of data. In the very special case, if we have no missing values, if we have measured quantities in every column, the conclusion from this type of linear model is identical to the conclusion of a two-step procedure where first we take a summary of all the features in a run and get one number per run per sample, and the second we focus only on the pink part and we kind of focus on the biological aspect. And this gave us an inspiration because these really big models, they're complicated, they take a long time to, to work with, and they have convergence problems, they fail in many ways. Figuring out that we could replace solutions to this model to a two-step procedure where first we do the summary and then we consider, essentially, first we take care of the yellow part and get one number per protein per run, and second we look at the pink part, allowed us to really become very general in terms of designs and in terms of experiments and handling missing values. So that's what we do. And it's interesting because, in many ways, this is what people do already, right? So Skyline would do exactly that, right? It will take the summary of all the features in a run and then do some other analysis, except that for us, understanding that there is this linear model behind this allowed us to go one step further and take care of missing values, outliers, complex designs, and so on. So that's where it came from. So let's kind of take a look into this. So the first thing we do, so we will focus on the yellow part, and we will ignore which run comes from which condition. We will only focus on the technology part, and this is where we will replace some of those values by NA censored, right? So now NA censored means that it was either missing everywhere except in Skyline, or it was a small value below our vertical line, and we replace the observed values with those NAs. And so now we write they're censored because we know they're informatively missing, right? So they have their values below the cutoff. And so now, in terms of censoring, we actually have ways. So there is, for this type of two-way table, it's essentially a two-way analysis of variance with censoring, where we fit a linear model which would allow us to account for the fact that some of these values are missing. And this is a very standard way of doing this. The problem with that is that it will take care of missing values. It will not take care of outliers. And so what we want to do now is to have this type of model which is simultaneously incorporates censoring and incorporates robust summarization. And there's again a statistical literature. Remember, I was saying we rely on libraries a lot in R, so that's exactly what happens here. We rely on a lot of work by others. And so here we have this summarization, which is simultaneously accounts for censoring and accounts for outliers. And this allows we can also impute values for those censored values. So this will be our estimates of what the missing values would be. But most importantly, the output of that is a new table which has one number per protein per run on some relative scale which is comparable between the runs.
When you're doing the imputation, do you have any sort of thresholding to make sure that you're not imputing these values based on, maybe one or two measurements out of 20, or, is there something?
Not really. So our constraint is, so let's say this value is missing, right? And we want to impute it, right? So the requirement is that we need to have at least one value for this feature in the experiment somewhere, and we need to have one value for this run somewhere across the features. And then we can essentially project. So the assumption behind that is that the features are supposed to go somewhat parallel. So if they're not parallel, it's viewed as random noise. And so if the value is missing, you say, okay, in this run, things went up. So I also expect this missing feature go up. Now this feature is lower than their friends, right? So in my imputed value, this feature will also be lower. And well, since you asked, let me tell you more details. So they're actually, in statistical literature, you can do a robust and censoring in one step. And we tried that. Turns out that it's more complicated and things fail more, and and we want it to be robust to many data sets. So what we actually do behind that is a two-step thing, where first we only do censoring without robustness, impute these missing values, and then we'll have a second round of summarization, which takes care of outliers. We just find that in practice it works better. And so if you have outliers, they will be taken care of as this in the second step.
But if you just need one value, does that mean you're potentially imputing almost an entire experiment's worth of data
Yeah. I see what you're saying. So we actually, that will be part of the converters actually. So we never led to the step features which have missing values almost everywhere. So the converters will have additional filtering steps. And in fact, I will talk about this just in a minute. So, and we have filters based on Q values of identification. You can have filters based on other criteria, right? What we also do is, well, yes, so you know, there may be some parameters that I may not be aware of at this point. So maybe there are some limits like you would not impute in certain cases, but at least conceptually, as long as you have at least one feature in a run and at least one feature, one number for this feature somewhere, technically you can do that. I think at the level of converters, you can implement your own filters, and we will see an example. I hope it will kind of help you see. And actually, that's exactly my point, that this type of steps matter quite a bit. So this cannot take care of everything, right? So what happens upstream and what Brandon and Alexei were talking earlier, it matters very much still, right? So we're not replacing all of the considerations that others. So let's come back to this. But so this part is important for two reasons. So one is if we want to continue talking about differential abundance, so now we will take this data structure, and now we will talk about, you know, time course, paired design, you know, and all of this, and we will get p-values. At the same time, if you want to do other things, for example, if you want to do machine learning, right, and do biomarker discovery and do predictive methods, or if you want to do clustering and draw heat maps, right, with proteins versus runs and so on, you can take these values as is, not do any statistics, and just use one number per protein per run, these summarized values, as input to machine learning or as input to clustering. So that would be another. So you would essentially have one row like this per protein. And this is something that can be done. And so the machine learning tools that we now have in MSSTATS, they would work with that. But going back to the differential abundance, so this is a little bit more details on the robust summarization. This is with Tukey median polish, which is a very standard method of doing that. Okay, so let me just kind of focus now on that. So now we will start interpreting, saying, okay, but some of these runs came from different conditions and different subjects, and this is where we look for the appropriate statistical model. And I don't remember how many models we have behind the scenes, at least a dozen, which will kind of account for different edge cases, and we will find the appropriate model which reflects the design. But again, from your perspective, all you need to do is to annotate which sample came from which condition, with the convention that each biological replicate has a unique identifier. Okay, so now I wanted to share with you some of the evaluation results, and first to show why we think that this type of approach contributes to reproducible research. So remember the beginning of the hour, I was saying that we want to make sure that our results are as little dependent on the choice of the tools as possible. So we want to make sure that our conclusions are not too dependent on the tools, and we looked into that. Okay, so the first one, this is DDA, unfortunately, but these are nice examples. So this was a controlled mixture where we had 4 conditions, and we had proteins spiked in different concentrations in different conditions. So we knew the true-false changes, but there was also a whole bunch of background proteins. So it was a complex mixture. And so this is an example of one protein. So this is what we call a profile plot, and MSSTATS generates a lot of those. Here the x-axis is the mass spectrometry run. The y-axis is log of the feature intensity. The light gray lines, these are the peptides of this protein in the background. This horizontal line is the line below which we do not trust the values. As you can see, for this protein, there's no problem. We trust every value, right? No missing values. Now, the green line would be the summarization similar to what Skyline would do, where we take the peak intensities on the original scale before log transformation, take the sum of everything for the protein, and then take the log. And then the red line is essentially MSSTATS proposed is the MSSTATS method. And then we have like low one is just linear methods, this classical approach, which is not taking care of censoring, not taking care of outliers. And this is Tukey median polish, which only takes care of outliers, but not censoring. But I think we can just focus on the green and the red lines. And you see that all of the linear models, they're kind of in the middle of the peptides, and the log of sum, so skyline-like summarization is above, which makes sense, right? Because we take the sum of all the values, right? So it will be higher values. So in this case, again, it's a spike in mixture. So this was a background protein, no changes. And you see that for all of these approaches, the estimated fold change is 1, the p-value is close to 1. So essentially, when there's no problem, you can do anything you want and you will be fine, right? Doesn't matter which tool you use. Now, here's a situation where we have interferences with low intensity features. And so still no problem, right? They're kind of above this threshold. And we see that, for example, skyline-like summarization log of sum is not really affected very much by interference among low intensities, because before log transformation, they contribute very little to the sum. So if you have interferences, it's okay. We can see here that, again, it's background protein, right? So the true-false change is 1, but the linear model, which is not robust, is affected by outliers. It has fold change 1.4. P-values are large, no problem, but it's a little bit affected already. But here's the situation when we have interference in high intensity peak, and so then it becomes a bigger problem, right? Because now the log of sum is very much affected by this interference, and you see this here again, background protein, true fold change 1, it reports 3.5. So this is a lot more, even though the p-values could kind of handle that. And of course, the real life, and especially with DIA, will look more like that, where we have a bit of everything, right? And we have low values, we have interferences in high intensity peaks and low intensity peaks. And so we see that here, this is a spiked protein, true fold change 7.5, linear model has 29 fold change, you know. The log of sum was too noisy, couldn't find anything, right? And so only taking care of both missing values and outliers helped us get to the results. So that's that, and this is really the kind of the figure I wanted to share. Mostly that we wanted to see if reading between the lines and processing these files in different ways will help us with the reproducibility, and the answer is that yes. And in every single data set we've seen so far, it helped. So these are different controlled mixtures that we have. So these are all DDA. So this is the one with 4 conditions I just talked about. This was the Cox LFQ, and this was the DDA spike-in, another spike-in experiment. So here we look for the number of differentially abundant proteins detected among true changes. And we know that they're true changes because it's a spike-in experiment, right? And so we see that if we only take the sum and then the log, the agreement between skyline, max quant, and progenesis is 11. It went up to 20 if we take care of these things, right? And across the board, right? So here it went to 32 from 29. Here we also had proteome discovery. It also went from 123 to 146. So in every single data set we looked at so far, not taking those values literally really helped reproducibility of the results. And for DIA, it's the same thing. So these are two data sets which we looked at. So this is spectral notice skyline, also increased the agreement between the two. So we believe that this type of approach helps us be robust to some extent to the choice of the data processing workflows that people use. However, and this is, and just to finish with that, the very last step, right? Once we have done all of that, what we had to do as part of this model fitting, especially on this stage, is to quantify the variability, person-to-person variation, technical variation if we have technical replicates, the extent of fold changes between the proteins. And because we have this information already, and because this is the necessary information to actually plan future experiments, we can use this to plan sample size of future studies. And as I mentioned last time, the number or the percentage of proteins which are expected to change has a big impact on the sample size. And so if we have only a small number of changes, it means that we need to work harder to protect ourselves against false positives. So we need a larger sample size. So MSSTATS will output plots that look like that. and you can do sensitivity analysis. For example, what we actually do here, literally, is that we have one variance component per protein. So to do this, to make this type of plots, we will take some representative variance, for example, 75th quantile to be on the conservative side, or maybe the median variance, right, for the protein. And for the fold change, we will take the smallest fold change we're interested in across all the proteins. So that is the idea. And so since then, MSSTATS grew into quite a bit of, quite a big project really, and quite a lot of code and a lot of collaboration. So we distribute this primarily through Bioconductor. So you may be familiar, it's a repository of packages for computational biology, but we also have a Skyline as an external tool as a Skyline external tool where we have a graphical user interface from Skyline, except that the functionality is somewhat limited compared to the base MSSTATS. So we have a quite active community of users. And so since then, it also grew into other things. And so what I cannot talk to you about today, but maybe you can check it out, is our recent work with TMT labeling as well for statistical analysis of TMT experiments. So now the next thing I wanted to mention, just to link it a little bit to the discussion that has been happening this week in terms of choices of data processing and also the impact, of the size of libraries or the number of features on the accuracy of the results. I think this is somewhat of a recurrent theme. So I just wanted to share with you the work which was actually led by Brendan and Mina Choi from my group primarily, but I think it is quite interesting to see how signal processing impacts the results. And the message here is that even though we do work very hard to take care of missing values and outliers, we cannot do everything. So we cannot replace entirely the work what is happening at the data generation and data processing step. So this is this experiment from Natalie Selevsek from the Abersold lab, which was profiling the yeast proteome by SWATH. And it's actually the same biological experiment as this 10 time point study I was showing earlier. So this is a response to osmotic stress, except that the original study was using SRM, and now this study actually is using SWATH or DIA. And it was a little bit smaller scale study, so 6 time points, 3 biological replicates per time point, and 18 DIA runs. And it was acquired on an instrument which is kind of lower resolution at this point, so triple top 5600. And Brendan and Mina wanted to look at the implications of data processing, how they propagate themselves all the way to the statistical analysis. And so here's just one example. So 2 analyses by Skyline. So one analysis used all the peptide precursors which were identified with the library and used top 6 fragments for these precursors. But the second analysis used additional information. So as part of the experimental design, the original authors of the paper, they actually first took 8 technical DIA runs from these different samples from different time points, and they essentially tried to understand how well they could quantify different peaks in the library. And based on that, they only decided to include in the actual study, in the second step, the peptide precursors which were not missing in at least 50% of the refinement runs and had coefficient of variation less than 20%. So that's kind of the answer of your question. So instead of saying, okay, can MSSTATS handle these extreme missing values? So what was done here is 2 options where one would just put in everything and deal with that, and the other is pre-filter things so that we have cleaner and better quality data. So this was one comparison, and the other comparison is looking at different data processing tools and how they deal with these situations. After that, we had exactly the same statistical analysis. So there was Q-value filter applied, and then we used standard MSSTATS for that. So this is the first summary. So we compared Skyline all versus low coefficient of variation. So in terms of number of proteins, clearly limiting your precursors, limited the number of proteins, right? It went from 2,500 to 2,100, so we lost roughly 400 proteins. And then here we compared essentially the progression of each time point, subsequent time point to 0. And this was the number of differentially abundant proteins identified by both analyses. Well, what we know from biology is that after the, and this biological system, by the way, was selected because a lot of biology is known about the stress, and so we would expect as time progresses, more and more proteins become involved, right, and become activated, and so we actually expect more and more proteins becoming upregulated in time. And so on the surface, we would say, okay, everything looks good because we identified more proteins, and if you just compare numbers, they're higher, right? So we consistently find more differentially abundant proteins. But then it's kind of suspicious because if you look at these 2 time points, going from 330, a very big jump to 500, but then it goes down and then it goes back up. So this is not something which we would expect biologically. If we only limit our data to low CV, we see that the progression is much more steady and stable. And you know, we don't know exactly what's happening, we don't have the ground truth, but to me this is more biologically plausible than this type of jagged situation, you know, with these many proteins. So, okay, we can kind of do more here. So for example, we can focus on this time point 60, which had like this big jump from 300 to 500 proteins in 30 minutes. And so we see that 275 proteins were found as differentially abundant in both, but there was some disagreement, and using everything identified more. And so we think that possibly this can be false positive. So if we look at the volcano plots, they look kind of similar, right? So there's really not much difference, but there is a disagreement between these two parts. And so what we can do then, just look at some of the individual proteins. And so these are again the profile plots. You will see a lot of them as part of MSSTATS. So this is a protein which is not changing in any of the analysis. So here the p-value is large, although here the adjusted p-value.
It's 10 o'clock.
I'm so sorry, I need to turn it off. So this p-value is borderline, right? So if suddenly you use the false discovery cutoff of 0.1 instead of 0.05, you can actually see this as differentially abundant. But so this is what happens here, right? If we just look at the individual features, we see that this type of filtering actually eliminated a lot of the things which we don't want to have, right? And so going back to your question, I think many of these features would be viewed as missing if we impose this cutoff, censoring cutoff that we have, right? And then it is possible that many of these features would be in a situation that you describe, and they didn't make their way to this type of filtered analysis. So at that stage, especially if you have many features which are missing a lot, I personally think that it is better to have our data more clean upfront as opposed to trying to deal with them statistically after the fact. This one survived, right? And so this is the one which will be truncated with MSSTATS and will be viewed as censored in MSSTATS most likely. So that's the idea. So again, we don't know what actually happens because it's a biological experiment, but just from the kind of our thought about the biology, this looks more plausible to me than this type of jagged pattern. This is another protein. So this one is one from this group, where here we found it differentially abundant after filtering, but not differentially abundant before filtering. And again, I think it's pretty clear that some of these noisy features contributed to much larger variation. But here's another thing what happened, and this is probably this is something that Alexei and Brandon already talked about. If you see here, all lost one replicate here. So actually, in this particular case, time point 0, when you have so many features, you also have to be a little bit more conservative with false discovery rate, and so the Q-value cutoff actually ended up losing one replicate in addition to introducing more variability. So in this case, I think after cleanup, we can probably say that the change is probably plausible here, and so this type of cleanup definitely is beneficial. Again, how did they do this? They actually had a prior experiment where they learned how much variability different features have, and they use this prior information to eliminate highly noisy precursors from the study. So they lost some proteins here, but I think overall the quality of the study improved quite a bit. And if you remember from Monday when we talked about the fact that these very long protein lists have negative implications for statistical analysis, right? So I think we can see the same thing here quite a bit. So this was one. And then the other is comparing different processing tools. So now we just stayed with the low CV version of the processing with Skyline, and also looked at Spectronaut and DIA Empire. And we looked at the number of differentially abundant proteins. And here is a zoom in into this time point 60. Now, clearly, there is a big difference between the tools. Let me give you first a disclaimer. So this is the older instrument. So, well, Alexei, I think he is gone, but he will tell you that 5600 is not the best instrument for DIA Empire. So DIA Empire works best with more kind of high resolution instruments. So I don't think it's a fair representation of DIA Empire in general, right? But the point here is not like comparing the tools, but saying that we do our best, right? But it doesn't mean that we can just put, you know, like all kinds of bad data in, and MSSTATS will take care of this. It will not happen, right? We really need to make sure that we process the data carefully, and then also use an appropriate statistical analysis later on. And so, again, which one is better? You know, Spectronaut kind of looks somewhat suspicious, right, with that many features, even though the increase is monotonic. It's kind of, numbers just, again, and I'm no biologist, maybe it's true, but to me it looks like this is probably too much. I think here it's clearly differences due to the instrumental, the property of the instrument. To me, this is probably the right middle ground here. And again, we can look into different proteins. So this is the same protein which was not changing at this time point. We were looking at it earlier. So Skyline has just this interference after filtering. Spectronaut, I'm not quite sure which filtering they used. They didn't, so this processing did not rely on these extra runs, right? So without that, clearly we have some noise. And I think DIA Empire in this particular data set had too many missing values to find things. And so this is the reason why we have discrepancy between the results.
So the comparisons you're talking about are all 3 versus 3, right? So they're extremely prone to noise.
3 versus 3 of what?
3
Oh, yeah, of course. Of course.
So what, in your graph that showed standard deviations were, you know, over, or what was in very small, was at some point you would have overexpressed.
Oh, you're saying that if I had more replicates, everything would be significant? That's what you're saying?
No.
But that's true, though, yeah.
The other one, the one with the p-values where you would get overexpressed differentiation with significant p-values. Anyway, that's fine.
Well,
Very small sample sizes are prone to having.
for sure. Oh, I see. Yes, so not exactly. So not only you don't find things, but if you do find, you may be exaggerating the fold change. Yes, definitely. So, but you know, there are many things here, right? But in this particular part, the point is that on one hand, we're very excited that MSSTATS allows us to handle some of this variation. On the other hand, my point is that things before we get to MSSTATS actually matter as well. So I'm not saying that we just can eliminate all of that. And this kind of is a bigger point, which I really would like to mention for kind of generally for statistical analysis. You know, for example, recently I had someone came up to me and said, you know, this multiple testing adjustment, it really doesn't work for me very well. So my p-values were small and looks good, but then I adjusted for false discovery rate and nothing is significant. So something is wrong with this false discovery rate adjustment. And I'm like, wait a minute. So you designed the experiment, right? You collected the data. You exported the data to whatever tool, right? So you processed the data, right? You summarize the data, missing values. You model the data, got p-values, adjusted. It doesn't work, and you blame the adjustment. So there are all these other things that we had to do, right, to get to the adjustments of p-values. And this is actually a very common situation. And these are, again, Jeff Leake and his colleague who even published a comment in Nature saying that p-value is not the ultimate goal, but p-value also is not the only point of scrutiny, right? And so what they talk about here is that we have experimental design, raw data, data cleaning, statistical models, summary of the models, p-value and adjustment. And the p-value oftentimes gets extreme scrutiny because, well, p-value is what keeps us from publishing the results, right, if they're not at the right level. But we really need to be very careful about everything that we do here. And the problem with all these steps above is that they're not really easily expressed mathematically, right? So t-test, we talked about this on Monday, you know there's a formula, you calculate the numbers, you get the p-value, you write a script, fine, right? But all of these things, there are many decisions that we have to make before we get to the final results, which are not necessarily documented the same way, are not necessarily quantified the same way, but they're just as influential for the results. And this is a known kind of thing in statistics. And, you know, there's a lot of kind of discussion about that. And here is a paper in Psychological Science, which says that undisclosed flexibility in data collection and analysis allows presenting anything as significant. So what does it mean? It means that we need to work with data sets like this, example data sets, control mixtures, to understand the limitations of different workflows, including the sample size, including the filtering, including data processing, and essentially establish the SOP, which we will follow is in the next study, and then stick to this SOP. And this, if this SOP gives us p-value which does not pass the threshold, well, so be it, right? Because we cannot really, if we start adapting our, you know, ways to filter things or ways to process things and have different parameters to get the small p-value, we will get the small p-value, but it will not be reproducible. And so this is a really important part. And so to me, this exercise really demonstrates how important it is to spell out upfront how exactly the data will be processed. And this includes, you know, all of this kind of quantifying, identifying peaks, filtering, and so on. So this is an important part. Okay, and now I just kind of the last part, which is actually not very long. I also wanted to talk to you about the things which essentially happened before, right? Before we get to the experiment, and how much we need to do before we even start designing the experiment and collecting the data. So I've been emphasizing the a lot the sample size, right, and the fact that we can measure things in a way which is reproducible. So there are two things which can be very necessary and very helpful, especially when we run large-scale DIA experiments in that. And so the first thing is understanding that our assay is actually capable of quantifying what we want to quantify. And this is kind of along the lines what the Selevsek paper was doing, where they first had a trial run when they said, okay, things which have too large coefficient of variation, we will not bother with that because it will inflate our dimensionality, but it will not be useful. Let's remove it upfront. So a similar thing that we can do is assay characterization, trying to understand, okay, for large-scale DIA experiments, what is the limit of detection? What is the limit of quantification? Does it give us what we want for the particular biological purpose that we have? And well, I know Tina will talk about this right after, but traditionally for low throughput assays, like for SRM or others, what you would do, you would essentially titrate the analyte at different concentration, make sure that this titration falls in the linear kind of range, part of the dynamic range, right? Then we will make plots with true concentration here and the intensity here, and we will make this straight line, and we will use it to determine limit of detection and limit of quantification. In DIA, the problem is that it's not possible to do this kind of very careful titration calibration for every single assay to make sure that it falls in the linear range. And so then problems start. So for example, here, this is actually a DIA peptide, right, from one such experiment. And well, you've seen these plots many times, right? And probably you would argue that this is a very good straight line fit. I would like to argue that this is a very bad straight line fit. And the reason is that we're not interested in this region, we're interested in this region, right? And so now if I zoom here, what I see is this. And this is because, you know, we have that many analytes in the DIA experiment, we could not really calibrate the assay for everything precisely. And so now we're fitting straight line to something which doesn't look straight at all. And it's a problem. So why is it a problem? Because, well, how are we going to determine limit of detection and limit of quantification? So what we will do is to, let's say we have a straight line fit of sorts that we fit to the data. We will also have some blank samples or some other representation of the noise in the spectra. So let's say here. And then statistically what we do, we build prediction intervals for the noise around the blank samples, and we also build prediction intervals for the noise around the titrated mixtures. And just to clarify that prediction intervals are different from the confidence interval, because they're intervals for the expected range of future data points, not just for the mean, but for the actual individual future data points. And then the point where the straight line intersects the upper prediction interval of the blank, we call it limit of blank, and when the lower limit of prediction interval intersects the upper limit of noise, this is limit of detection. And so obviously the location of the straight line determines LOB and LOD, and so if we have fit the straight line incorrectly, we will miscalculate LOB and LOD. And this is actually what really happens in practice. And so when we have situations like this, it turns out that the straight line is actually tilted with a lower slope, meaning that our figures of merit are too small, so too optimistic, and the assay is actually not as good as it looks because our straight line was not fit correctly. So what we did here for this purpose, to assist those who use DIA experiments and trying to understand if this is actually a good tool for their purpose, we instead try to express what we call a canonical calibration curve, which really says that, well, you know, if you have concentrations which in your study which are below the limit of detection, your true curve is actually this hockey stick pattern as opposed to straight line, where you have some part in the linear part region of the dynamic range, but some part is below the linear region of the dynamic range. And this, by the way, is the collaboration with Mike McCoss and Jared Egerton and the Skyline team. And so what we have done here, so now we have this curve, so instead of fitting a straight line, we want to fit this kind of bilinear line. But the problem is that this change point is uncertain, right? We don't know where the change point is. And we also want to make sure that we can characterize the location of this change point. And so the way we do it, it's a fairly standard statistical procedure called bootstrap, where we randomly sample a subset of the data points here, feed those bilinear curves to each of those sampled subsets, and if it fits completely straight line, it's straight line. If it fits bilinear line, it fits a bilinear line. And then what we do, we average over all of those bilinear line fits. And it gives us something which is non-linear, somewhat smooth, not quite sharp as the original curve, but it reflects the uncertainty in the location of the change point. And so now we have the prediction intervals which reflect both the curvature and the uncertainty in the change point, and in a way which is very robust to any type of configurations, you know, of the titrated samples. Now, if it turns out that you have an experiment which is entirely in the linear dynamic part of the dynamic range, you will have the identical straight line. So it essentially extends what the linear line is doing. And so what turns out is that the real figures of merit are often more conservative than is reported from the straight line. And so, well, in real life, we don't know the truth, so we did some simulations where we simulated data like that. So essentially, if you fit a straight line, it looks like this. The slope is smaller, and therefore, the figures of merit are more optimistic. And so in simulation, we found that it can be too optimistic by as much as 60% compared to the true values. And our values are a little bit too conservative, but just a little bit. So here is the percentage of error, and the linear models for limit of blank and limit of detection can give as much as 60% of error on the optimistic side. And this is a whole collection of real-life calibration experiments. We have a DIA, PRM, and a whole bunch of SRM experiments. And we don't know the truth, but we compare our values to the values from the linear curve, and in the majority of cases, our values are more conservative. So we think it's something that we need to make sure that we pay attention to. So this is one. And then the other, very last one is the fact that we talk a lot about large-scale experiments, right? That we need to have more replicates. But if we want to have more replicates, it also means that our measurement system has to run consistently and performs in a way that is sustainable for many runs. And so we came up with a series of statistical methods which help you do two things. So one is longitudinal monitoring for system suitability, meaning that before collecting the data, we can monitor standard mixtures to see that the measurement system performs as required. And then also quality control. These are the standards that you profile kind of more frequently to ensure that when you collect the data, the system performs as expected. So there are a lot of kind of fairly standard tools for that. So, and you know, traditionally in statistical literature, people use control charts. So for example, here X chart looks at, in this case, you look at the mean of the retention time in this, or here's peak area. So this is the mean of a peak area in time. And essentially what these methods require is that you will give an example of 10 runs or 15 runs, which you manually verify are good quality runs. From these runs, we learn the extent of acceptable variability of these standards, and we would learn the thresholds. And then we will plot, for example, peak area here, and as soon as the peak exceeds the threshold, we say that this may be a problem. So this is kind of a simple case, but then also we can have standardized versions of this, and this would look for changes in mean signal, like peak area, retention time, peak asymmetry, you know, any type of quantities that you're interested in. The problem with this is that sometimes deviations are different from just shifts in means. What can also happen is the shifts in variability. For example, in retention time, one problem with retention time going kind of, you know, not as expected, it's not like it's drifting, but it's becoming more variable. And so you also want to be able to detect changes in variation in addition to changes in mean. The thing is that it requires different statistical summaries to detect changes in variation. And we can also look for isolated signals, we can look for small drifts, and we can also find the points where the changes, the earliest time points, the changed point time, where these deviations occur at the earliest possible time. And so the problem is with this, that if you start looking at multiple peptides with DIA, right, and each peptide has multiple metrics, peak area, retention time, peak asymmetry, peak width, and each of these metrics can have different types of problems for which you need different statistical summary. For the mean, for the variance, for the drift, you know, for outliers, you get a very, very, very large number of control charts. And then, you know, it's very hard to tell what exactly is happening. So the current iteration of these tools provide essential graphical summarization of these results. So this is the most high-level view. So here the x-axis is the time points at which the standards are monitored, and the y-axis would be different metrics. For example, here for the mean signal and peak area and retention time. So blue means that the majority of the signals are in control. Orange is some proportion of signals are out of control, and red means the majority of signals are out of control. So you can just look at the first glance and see how things are going. Now, if things get orange or red, then you can look at these plots and say, okay, where exactly the things go wrong. So this type of plot summarizes, so looks across time and summarizes across peptides. So here, different color represents different type of problems. So I think yellow here is the change in the mean. So you see that here, this is the proportion of peptides which start having change in the mean. This is for retention time, and here it will be change in the mean, and then so going up in the mean, going down, or change in variance. Here there is no change. So it allows you essentially to summarize over peptides in time. Here it summarizes over time, but for individual peptides. So here each point on the circle is the peptide, and this is the proportion of runs where this peptide had a problem of any kind. And so you can also see individual peptides, what's going on with them. So this is the current implementation of this tool, but we actually have a very recent new iteration on that, which is in the beta version, but we will release it very soon, where we do more than that, but we actually use machine learning tools to make predictions of in control or out of control for the entire experiment, and we can have error rates of prediction summarizing across all metrics, all peptides, and all types of problems simultaneously. Okay, so I guess I was so impressed last time that I went over time that I'm finishing a little bit earlier today. So let me just have some summaries of what we learned as part of this process. And again, from the perspective of contributing to reproducible research. On this kind of yellow side, right, kind of minimal standard of reproducibility, we want to provide scriptable analysis which have documentable automated reanalysis workflows. And here we work with fully transparent algorithms and code which can tell you how we handle different types of tools, and we can still kind of try to interpret them in the way which is the most consistent. And of course, here we work on the data analysis kind of from the modeling for reflecting the experimental design appropriately, but also making sure that our measurement system is as expected and also doing assay characterization. And here, this is more like the Monday topic when we talk about sample size calculation, but also selecting the right replicates and designs. And really, for this part, SOP for the entire design and analysis is really key. So I just wanted to thank very many people. So first of all, in my group, many people were part of these different aspects of this project. So Mina is the main leader and spirit behind the whole MSSTATS work. Eralp contributed a lot on the MSSTATS QC part. Ting is working with TMT, but also her work on protein effects of protein number of prediction that I talked about on Monday. Cyril was working on LOD and LOB determination. Sarah contributed code to the graphical user interface for quality control, and Sung-Hen, who is here, will be co-leading tomorrow the hands-on part, worked on things actually I haven't had much chance to talk about, such as post-translational modifications, features which don't agree with the majority of features, and so on. And a lot of work from the controlled mixtures came out of the ABRF IPRG group. And of course, a really important part of this is the collaboration with the McCost lab, in particular Brendan and Jared, and of course the ETH lab as well. And many of the ETH members are beta testers of our tools, and we're very grateful for that as well. Okay.
For Olga. Yeah.
Going back to the normalizations,
Mm-hmm.
right? Sort of, right at the start with the using the standards. So when you're talking about standards, are you talking about like a heavy version of your peptide of interest? So if you don't have the spike-ins, basically you have to do the sort of median or quantile? Is that?
Well, you have the intermediate approach where you just have one or two standards, not a standard for every peptide, but just one or two global standards spiked into your sample.
So if that's the case, my question sort of is, how early do the standards need to be spiked in to ensure that you're not just measuring pipette error?
As early as possible, yeah. Well, ideally, you would have some endogenous proteins which are very tightly regulated and don't change. I've never seen such a thing, but that would be ideal, right?
But you can use endogenous.
Oh yeah, if you could, if you know in your kind of biological setting, you know that something, oh, those are the best because those account for everything which happened to the. It's hard to know those, but if your specific setting has those, that's by all means use those.
But
also. Yeah.
Oh, it's just, maybe I can, if we're now talking in the DIA setting where we have a lot of information, do you think that the sort of global median or quantile approach is going to be more stable than this type of spiking you're talking about, or have you looked into this?
Well, yeah, so it's really, so see, the way to think about this is that every normalization makes assumptions, right? And then you say, okay, what are the least kind of stringent assumptions that I can make in every case, right? So having your proteins not changing, and those proteins may have like multiple peptides and multiple fragments, so that's really a good way to go, right? If you don't have that, then you say, okay, if I have an SRM experiment with 20 proteins and all of them are in this pathway, which is supposed to be affected, you don't want to do quantile normalization, right? So then what, right? So then having some way of using some external normalization. If you can target a couple of proteins which are not changing, that's best. If not, spike in references to control, like for different digestion and so on steps. What we also kind of see increasingly people use IRT peptides for normalization, right? But it's also silly because I noticed that some people don't spike IRT peptides in constant concentrations, and then it's kind of lost. So if you think about this upfront, then you can actually use IRT peptides if it's done carefully.
Is there a reason why you didn't implement other, besides just obviously the difficulty of coding it and those sorts of things, but sort of why you didn't go for something like RUV or like an MLR type normalization? So like a...
Which ones? So what are those?
So RUV, I can't remember what it stands for, but it does a similar sort of thing to the sort of standard-based normalization, except it looks at large number of endogenous, and it is more forgiving for changes.
You know, it has not been necessary so far. So we try to keep it simple also, right? And so for the moment, we have either single standard or constant normalization or quantile normalization, right? So far it worked, but the more complicated it is, you know, the more opportunity you have for it to fail and not notice, right? So I would say we try to do good enough, but for many studies, as opposed to go in depth, but then realize that it may be overfitting or overdoing something in some cases. Yeah.
Other question, Tina?
For this data set from STUDY Selevsek, you said that there was a pre-experiment done with 8 samples to find the features that are most varying. But with DIA, couldn't we have done it directly on the data? Why was it necessary to do an experiment for additional pre?
Well, I think that was also part to experiment with the DIA settings and all of that. So I think that was part of just kind of setting up the assays, I think, because it was a while ago, right? So there was more kind of work to do. But you're right. So actually, and this is something that you could do, and this is called non-specific filtering. The most important criteria there is that you do not use the information on biological conditions. Right, so essentially you cannot use the same data twice, once to filter, right, and then to do statistics on this. So it has to be external. Non-specific filtering means it doesn't use the condition on the information on conditions. So for example, if you have a very high CV, maybe 20% is too low, but let's say 40 or 50%, right? You can say, I'm removing every feature which has CV more than 50. Can you remove true biological variation? Sure, you can remove some real interesting proteins this way, but you know, at least you spared money and didn't run these extra experiments before. You can also say, I'm removing features which have CV very low, because it's probably because they're reaching the saturation and they don't vary enough, right? And so those you can also probably remove. You can remove things for other reasons. For example, I don't know, I identify the protein, I don't know what to make of this protein, right? I cannot follow up on this protein. I eliminate it upfront before even doing statistics, because it will not be useful to me. So any criteria you can use, provided that the same data are not used twice. So what Brendan is also suggesting is to have technical replicates and then use the CV of technical replicates, because then you don't use biological information, but this means that you will have technical replicate DIA runs, which also may cost extra money. So these are trade-offs, I think, but I think the bigger point here is that the more prior information you use, the better your statistics becomes. It's a fact of life in general statistics, right? So the more prior information you use, the better you do, right? And so what I've seen, especially in earlier days, people say, you know what, these days computation is so powerful and fancy, let me just throw all the data at it and see what happens. Not much happens, right? It's really good to put prior information in as much as possible.
So I think if you try to think...
But so really the important points are to eliminate proteins which are not serving you, or features which are not serving you upfront will help. And the other is not use the same data twice, because otherwise we overfit.